In the name of Almighty God, I am Master Hassan Kashi, and today I want to talk about protein adsorption. Protein adsorption is the process where proteins adhere to a surface, often a biomaterial, when they come into contact with a fluid like blood or solution. This process is influenced by the properties of both the protein and the surface, such as electrostatic interactions, chemical properties, and topography, and it can affect cell adhesion and biomaterial performance. It's a crucial event in applications like medical implants and diagnostic devices, and its outcome can be either beneficial or detrimental depending on the specific use case. And what are the key characteristics? The first one is immediate occurrence. Protein adsorption is often the first event to occur upon contact of a biomaterial with biological fluids, happening within seconds. The next one is surface and protein properties. The process is governed by the physicochemical properties of both the surface, for example, hydrophobicity, and chemical composition, and the protein, for example, size, charge, and affinity. The next one is cellular interactions. The adsorbed layer of proteins on a material surface can influence subsequent cell adhesion, proliferation, and biological response. The next one is conformational changes. Protein can undergo structural and conformational changes upon adsorption, which can alter their function and expose new sites, such as epitopes, that trigger a specific cellular or biological response. The next one is thermodynamic driving force. The process is governed by fundamental thermodynamic principles seeking a state of lower free energy, which is driven by factors like the reduction in solvent accessible surface area and changes in entropy. And what are the applications and implications? Protein adsorption is a critical factor in the biocompatibility of implants, gathers, and other medical devices. It can lead to issues like membrane fouling in dialysis and affect blood cell interactions. The next one is diagnostics and drug delivery. It is essential for many diagnostic tools such as microarray and lab-on-a-chip devices where precise immobilization of proteins is needed. In drug delivery, it can be used to target nanoparticles to specific cells or lead to their inactivation. The next one is industrial processes. Protein adsorption also happens in other fields such as the food industry, where it contributes to the formation of scale in equipment like beer kegs and tap lines. Adsorption is a process where molecules, ions, or atoms adhere to a surface, unlike absorption, which involves molecule permeating the bulk of a material. This surface phenomena happens when a gas, liquid, or dissolved solid comes into contact with a liquid or solid surface, known as adsorbent, forming a film of the substance being attached, called the absorbate. Adsorption is a surface-based process and is often used in purification, separation, and catalysis, with applications ranging from water filter to gas masks. And what are the type of adsorption? The first one is a physiosorption. Relies on weak intermolecular forces such as van der Waals or electrostatic forces. It is reversible and similar to condensation, and the example is water vapor condensing on a cold glass surface. The next one is chemisorption, involve a chemical reaction where the adsorbate form a chemical bond with absorbent's surface. It is much stronger bond than physiosorption, and the example is water molecules breaking apart on a metal surface to form H and OH bonds with the metal. And what are the key characteristics and applications? The first one is exothermic process. Adsorption typically releases energy, making it an exothermic process. The next one is temperature dependence. Lower temperature favor adsorption because it is generally an exothermic process, and increasing temperature can favor adsorption. The next one is surface area. Materials with a large surface area like activated carbon or silica gel are highly effective adsorbents. And what is about applications? The first one is purification, removing impurities from liquids and gases, such as removing odors or decolorizing sugar solutions. The next one is separation, separating different types of molecules, like in chemio, uh, chromatography. And ion exchange is a powerful method to separate protein from solutions and is today readily used in a biotechnological and pharmacological industry. This model simulates an ion exchange column for protein adsorption. The fluid phase contains four components: two proteins, solvent, and one salt. The adsorption desorption kinetics is described by two equilibrium reactions where proteins displace ion absorbed at the surface and vice versa. The example highlights how reactions at chemical equilibrium can be studied in a zero-dimensional reactor system in a reaction engineering. In addition, it also shows how the kinetics from the zero-dimensional setup is exported to a three-dimensional model where the reacting surface in the column can be studied in detail. The three-dimensional model incorporates mass transport through diffusion and convection, and the reactions at the surface of the ion exchange mass with transport of the diluted species, a free and porous media flow, and surface reactions interfaces. It has been shown that a protein molecule adsorbing to a solid surface with immobilized alkali resides will bind multivalently to several residues at the same time. The contact points with alkyl residues can be considered as individual binding sites situated in either a homogeneous or a heterogeneous two-dimensional lattice. In principle, therefore, the amount of the absorbed protein under standard conditions also depend on the surface concentration of binding sites on the solid phase. On the basis of multivalence, which implies a specific geometry of sites, protein adsorption on such tailor-made lattice has been compared to a molecular recognition process. Terminologically, just as a coenzyme is viewed as being a small entity, for example, a ligand in binding to a macromolecular enzyme, a protein can be similarly viewed as being a small entity. For example, a ligand in comparison to the large macroscopic solid surface it is being adsorbed to. For simplicity, therefore, protein molecules in protein adsorption studies, adsorbates have been defined as ligand. To account for the two fundamental parameters governing protein adsorption on natural and artificial surfaces, one may distinguish two categories of protein adsorption isotherms. The first one is lattice site binding function, and the next one is bulk ligand binding function. Equations describing these binding functions have been derived previously and are similar power functions as is the Hill equation. Proteins are interfacially active molecules, a statement that is demonstrated easily by the spontaneous accumulation of protein at interface between 1 to 4 as a reference. While the proteins show the propensity to absorb the interfaces, and why do they adsorb too tenaciously? For some proteins, the tendency to adsorb is due to the nature of side chains present on the surface of the protein. Protein is amphoteric polyelectrolyte. Its amino acids have different characteristics. Some are apolar and like to be buried inside the protein globule, whereas others are polar and charged and are often found on the outside protein surface. A strong long-range electrostatic attraction between a charged absorbent and oppositely charged amino acid side chains will lead to a significant free energy change, favoring the adsorption process. In other cases, the interfacial activity of the protein may be driven by its marginal structure stability. The compactness of the native structure of the protein is due to the optimal amount of polar amino acid residues. The stability of such a structure depends on the combination of hydrophobic interactions between hydrophobic side chains, hydrogen bonds between the neighboring side chains, and along the polypeptide chains, and the Colombo interaction between charged residues and van der Waals interactions. An adsorbent surface can compete for the same interactions and minimize the total free energy of the system by unfolding the protein structure. The adsorption process may result in a surface-induced protein denaturation. Elements of the secondary structure of the protein, alpha as helix and beta is sheet, together with the superconductory motifs from form a compact globular domain. Some proteins are built from more than one domain. In a multi-domain proteins, it is possible that one domain will dominate the adsorption property of the whole macromolecule at a particular type of interface. For example, acid pretreated antibodies bind with their constant fragments to a hydrophobic surface. In order to completely characterize and predict protein adsorption, one would like to have a quantitative description of the adsorption. This description is typically obtained by measuring the adsorption and desorption kinetics, conformation of adsorbed proteins, number and character of protein segments in contact with the surface, and other physical parameters related to the adsorbed protein layers, such as layer thickness and refractive index. This simulation describes a selected set of techniques and protocols that will provide answer about the mechanism of protein adsorption onto and desorption from the surface. The reader is referred to specialized monographs and a review here on protein adsorption for a more comprehensive coverage of various aspects of protein surface interaction. And the description of protein adsorption. The first one is adsorption isotherms. The adsorption isotherm is a function that relates the measured adsorbed amount of a protein per unit area to the solution concentration of protein, typically increases sharply at low solution concentrations of protein and levels of at higher protein concentrations approaching a limiting value. The existence of tau p adsorption or plateau has been interpreted as a sign that the adsorption surface is saturated with protein molecules. Any further increase in the solution protein concentration typically doesn't affect the TPU. The amount of absorbed protein in the plateau of the adsorption isotherm is often close to the amount that can fit into a closed-packed monolayer. Hence, the notion of the saturating monolayer coverage is often applied to protein adsorption. Under ideal conditions, the shape of the adsorption isotherm can provide information about the affinity between protein and surface. The evaluation of the affinity, however, does require a model for the protein surface interactions. A model from the which the absorption isotherm function can be derived and compared with the experimental results. Most experimentally measured protein adsorption isotherms may not be the true equilibrium isotherms. It may take a relatively long time for a protein adsorpting to a surface to reach true equilibrium. Other processes such as protein conformational change may run concurrently with the adsorption process and affect the adsorbed amount. Hence, the information contained in the adsorption isotherm may not refer to identical molecules. The existence of several protein conformers in solution, each with a slightly different adsorptibility, may result in an isotherm that will reflect the competition between conformers for a limited adsorption surface area. The adsorption to two-dimensional lattice of binding site. It has been shown that a protein molecule adsorbing to a solid surface with immobilized alkyl residues will bind multivalently to several residues at the same time. The contact points with alkyl residues can be considered as individual binding sites situated in either a homogeneous or a heterogeneous two-dimensional lattice. In principle, therefore, the amount of the adsorbed protein under standard conditions also depends on the surface concentration of binding sites on the solid phase. On the basis of multivalence, which implies a specific geometry of sites, protein adsorption on such tailor-made lattices has been compared to a molecular recognition process. And terminologically, just as a coenzyme is viewed as being a small entity, for example, a ligand in binding to a molecular enzyme, a protein can be similarly viewed as being a small entity. For example, a ligand in the comparison to a large macroscopic solid surface, it is being adsorbed for. For simplicity, therefore, protein molecules in protein adsorption studies or adsorbate have been defined as ligands. To account for the two fundamental parameters governing protein adsorption on natural and artificial surfaces, one may distinguish two categories of protein adsorption isotherm. The first one is a lattice site binding function, and the next one is bulk ligand binding function. And the equations describing these binding functions have been derived previously and are similar power function as is the Hill equation. And here you can see in the component section that I have to use the transport of diluted species and surface reactions and laminar flow. And here is there is a reaction engineering. And if you can go to it, you can see the species and the formula and equation. And also there is a velocity field as a contour, a bulk concentration, and surface concentration. And thank you so much for watching my video until the end. And also you can download my tutorial video from github.com/masterskill.